In this video, I'm going to be talking about G-Protein Coupled Receptors, or GPCRs. These receptors are highly varied and control most cell responses to external stimuli in the cell. Their name comes from the coupling of a receptor and a G-protein. G-proteins are heterotrimeric, the alpha, beta, and gamma subunits that are bound together at resting state. Different GPCRs have different G-proteins and therefore different targets. The receptor's two main parts include the transmembrane single polypeptide chain folded into a globular shape that passes through the plasma membrane 7 times. Beneath the plasma membrane inside the cell, the G-protein with its subunits are bound to the receptor and the inner membrane. At its resting state, the alpha, beta, and gamma subunits are bound together with the gamma subunit attached to the plasma membrane and a GDP molecule bound to the alpha subunit. When a ligand, that could be peptide hormones or neurotransmitters, bind to the receptor, there is a conformational change in the polypeptide chain. This GDP on the alpha subunit is then exchanged for GTP. Then, the G-alpha particle dissociates from the beta and gamma subunits. Both the alpha and beta-gamma subunits are then useful in relaying of messages. It can target enzymes and ion channels. RGS regulates when the G-alpha hydrolyzes GTP back to GDP, where we go back to resting position. Here is an example of a GPCR that opens an ion channel. This is in the heart, ion channels in pacemaker cells of the heart, where cells can spontaneously depolarize and contract. Acetylcholine binds to the receptor, which swaps GDP for GTP. The GPCR is now active, and the alpha particle dissociates from the beta-gamma subunit. The beta-gamma subunit migrates through the plasma membrane towards potassium channels. Potassium leaves the cell, depolarizing it, which then leads to action potentials and muscle contraction. The alpha subunit is doing other functions. This continues until the alpha subunit hydrolyzes GTP back to GDP, which brings us back to the inactive state. This is a very rapid association, GPCRs with ion channels. In other cases, the alpha subunit associates with other enzymes and creates a second messenger that can interact with effector proteins that spread throughout the cell and influence it. Two of these enzymes are Adenylyl Cyclase and Phospholipase C. Here's an overview of the GPCR cascade as the G-alpha subunit interacts with Adenylyl Cyclase. First, epinephrine binds to the receptor. The alpha subunit will dissociate from the beta-gamma subunits and go interact with Adenylyl Cyclase. This cleaves off 2 phosphates from ATP, creating CAMP, which is cyclic AMP. Our second messenger, CAMP, then binds to the regulatory units of an important enzyme called protein kinase A, which is usually inhibited. These regulatory units are pulled away from the catalytic units. These catalytic units are the parts that do the enzymatic work. This C goes and finds normally inactivated phosphorylase kinase, phosphorylates it. Glycogen phosphorylase is then activated by phosphorylase kinase, and then this goes into glycogen stores and starts cutting it, creating glucose for ATP production. In another case, if a hormone is the ligand, then we get something different. The protein kinase A is activated, its catalytic unit goes into the nucleus, and changes transcription behavior, either inhibiting or activating genes to be transcribed. These genes are usually inhibited by CREB, which can be phosphorylated by pKa to be activated and transcribed. So we can see that we first start with many inactivated enzymes or proteins, but once epinephrine or hormones bind to our GPCR, this creates a whole cascade that introduces our CAMP, the second messenger, to create change throughout the cell. In this next example, our alpha subunit interacts with phospholipase C, or PLC. PLC will create 2 second messengers by cleaving an inositol phospholipid, giving us inositol 1,4,5 triphosphate, or IP3, and it will also give us diacylglycerol, or DAG. IP3 will bind to ion channels in the endoplasmic reticulum to release Ca2, or calcium. DAG will stay in the membrane and find PKC and activate signaling pathways. If acetylcholine acts as the ligand, phospholipase C can secrete amylases and aid in digestion or contract muscles for movement. If thrombin is the ligand, aggregation is the target to help with blood clotting. As for IP3, calcium is very necessary in the reproduction and fertilization, where calcium release is very important to prevent polyspermy. This is seen as soon as the sperm fertilizes the egg, a wave of calcium spreads and envelopes the eggs, pushing off the extra sperm. Calcium can also activate calmodulin and activate a host of other cell responses. Overall, the GPCR is a highly varied type of receptor that causes very impactful changes in the cell. It begins as a polypeptide in the plasma membrane connected to a G protein, which, when activated, will go and create second messengers that will create more cascade responses in the cell, leading to many things such as muscle contraction, heart rate regulation, gene regulation, and many other things. I hope this was helpful. Thank you.